Okay, I'm Dr. Jeff Stratford from Wilkes University. This lecture is Conservation Genetics. It is the last lecture I'm going to record for fall 2020. And it is such a relief. Okay, let's go. First, let's just define what conservation genetics is. It's applying genetic principles to promote conservation. And this could include studying gene flow, looking at adaptation, looking at genetic diversity, or species discovery. So it's a very broad term. All right, genetic variation. When we look at the hierarchy of life and the possible ways to measure biodiversity, uh, most measures of diversity look at species diversity, and then you have diversity of, say, ecosystems and habitats. Uh, if you go below the level of species, then you get into genetic diversity. So within a species, there can be diversity, and that would be genetic diversity. It's a measure for adaptation. So in the short term, it could do things like give a species disease resistance. In the long term, it can mean adaptation to changing conditions. Mechanisms that produce genetic variation. It depends on which level you're looking at. So at the level of the gene or the locus, you have allelic diversity. So that's the if you look at a population and look at the alleles that are out there, um, allelic diversity is how many alleles are out there for any particular locus. And when you do something like a Punnett square and looking at Mendelian genetics, a real simple model just uses two alleles for a locus, big A and little a. But the reality of it is, you can have dozens of alleles out there for any locus. So what produces, where do these different alleles come from? Well, the sort of the raw material of this diversity is mutation. And that includes all organisms. And what I mean by all organisms is that it doesn't matter if you're bacteria or a bird, that mutation is the raw source of genetic information. For sexually reproducing diploid organisms, genetic recombination is the source of allelic diversity in such a way that when there's crossing over, that the um, alleles for any locus for the homologous chromosomes that are crossing over, um, that it's broken up and exchanged, and you can get genetic information. I mean, genetic diversity that way. At the level of the population, most scientists look at heterozygosity. And so, you know what it means to be heterozygous? That means you have, in a diploid organism, you have allele from the mother and one from the father. And if they're different, they're heterozygous. And if you're saying they're homozygous, regardless of their identity. If they're just different, it's heterozygous. And so genetic variation is measured by the number of heterozygous individuals out there. Um, so things that promote genetic variation at the level of the population would be things like outcrossing and gene flow. And also at the level of natural selection, disruptive selection. Disruptive selection is when you are actually selecting for diversity. So some examples I know of is selecting for very large males and then very small males, because each has a strategy for mating, and this is with salmon species. And so disruptive selection means you're preserving this gene and this gene, and so you are maintaining that genetic variation. And outcrossing means you are breeding with individuals that are not related. Okay. There's also a spatial element to genetic diversity. And so I'll give you two scenarios, kind of extreme, where the level of heterozygosity in one is 0.31 and the other one is 0.79. And this arises because even though I kept allelic diversity the same, so this species in both scenarios has four different alleles, A, B, C, D, and what you get is spatial organization in the left, and that can actually erode genetic variation. And in the other, you have panmixis, so everybody's breeding with each other, and you can maintain genetic or heterozygosity, right? So allelic diversity and heterozygosity are two different things. And just because you have allelic diversity doesn't mean you have heterozygosity. And flip to that also. Other things that erode genetic diversity, stabilizing selection. So stabilizing selection means you're not selecting for the big ones or the small ones or the tall ones or the short ones. You're selecting for the average. And what happens is in a phenotype that is driven by multiple alleles or polymorphic DNA, you'll erode away those alleles that pull it to one of the extremes. And basically, you're just selecting for a particular set of DNA, a particular allele, I should say, that has highest fitness. Okay. Also, small populations erode genetic diversity. So things like inbreeding results in the loss of alleles. And the random loss of alleles due to genetic drift. And that's particularly important in small populations. Population bottlenecks are situations where you have a large population, it gets small for a little bit and expands again, but while it's small, you lose and shed a lot of alleles. And that can have actually benefits as well as disadvantages. So the disadvantage is that you lose genetic diversity. The advantage, well, most people don't think about this, is you can actually get rid of rare deleterious alleles. So through mutations, you can get alleles that are deleterious. So most of these are recessive. And what happens is if two recessive alleles get together, right, then it forms a phenotype that lowers fitness. And the lowering of fitness for one may lower the fitness for everyone. And these rare mutations can accumulate in a population. And so at any one time, two individuals getting together will have some gene that has these rare deleterious recessive alleles getting together and lowering the fitness. So the fitness of the entire population can go down. What happens in a population bottleneck is that when it goes down, you lose a lot of these rare, they can be selected out or just lost randomly. So there may be actually advantages to having going through a population bottleneck. In general, though, it's considered to be not good for the population because you lose the good as well as the bad. All right. Inbreeding depression is lower fitness um, as a result of breeding with close relatives. There's a loss. There's a loss of genetic diversity, and you have increased probability of homozygous recessive genotypes with deleterious effects. And what that means is um, you have rare recessive alleles. They're kind of spread in a population, and the chances of them meeting a large population are pretty low. But as the population gets smaller, or you start inbreeding, that the chances of those two recessive alleles getting together is much higher. This plant, by the way, with the flowers is coffee. It has beautiful flowers, makes a beautiful, delicious fruit as well. Okay, so small populations can go through bottlenecks. They can have all sorts of negative consequences, increased inbreeding. Um, but you can't just look at the population number per se. You have to look at the effective population size. And that's because you can't look at the population as if everybody has an equal chance of breeding. Okay? Um, what you have to look at is of, say, 100 individuals, how many of those are actually going to contribute their genetics to the next population? And that is the effective population size. What this graph on the right shows is how much variation is left behind after 10 generations. So if you have an effective population size of 1,000, basically you are able to maintain all of your genetic variation. Below that, you start to decline. And what happens is the smaller your effective population size, the more rapidly you decline. So you can see that if you start with five, you know, five individuals, which is really low, that you end up having only 40% of your genetic variation only after 10 generations. So this is not good. So for example, California condors got to 20, so they're going to fall after 10 generations. They're going to be somewhere in between, say, 65 to 90% of their genetic variation left, and probably closer to 60%. So there's inbreeding depression, and so the thing that would offset that is outbreeding. But there's also outbreeding depression. And so the issue you have is that you have a situation where if you outbreed, you can actually lower your fitness. How does this happen? Imagine a widespread population and there's local adaptation. So I put a sloth here, and I have no idea if this is actually true, but there seems to be a population of sloths that like mangroves and the coast, and then there's other individuals that like, say, regular rainforest, not the coast. So not mangroves, not the ocean, but terra firma forest, so dry forest. Now imagine that there's some sort of genetic adaptation that allows the sloth to have higher fitness in mangroves, and in the other population, they have higher fitness in the forest. And if you take sloths and transplant them, they have lower fitness. So if that's the case, then not if you transplant the individuals, but if you have gene flow. So if a male and female sloth meet up and have an offspring, it would either be adapted to the mangrove or to the forest, or possibly neither. In that case, it has lower fitness. So In the extreme, uh, outbreeding depression would be the more distant you are relatedness, the lower the fitness is. And so if you're a different species and you are sterile, then you have the lowest fitness, right? Zero. You leave no offspring. If you breed and have with a different species, but maybe related species, and you can have offspring, but if it's sterile, that's not good. So that is outbreeding depression. So if you can imagine, I don't know if I can do this here. Let's see. Can I annotate? Yes. So let me draw here, and this will be fitness. Okay. So I'll just say fit. And this is relatedness. I'll just put relate to move this along. What you usually get is like a hump shape. Okay. Where this side, so if you're not related at all, then you have outbreeding depression. And on this side, where you're very related, you have inbreeding depression. So what this suggests is there's some range in between, and this would depend on the species, would depend on the genetics. There's some range in between being very unrelated and being too closely related, where your fitness is at a peak, okay, where you don't disrupt your adaptive suites. So adaptive suites would be a whole bunch of traits that are adapted for that local condition, so you're not disrupting that. And yet it's different enough that you don't have inbreeding depression. Oh, and that picture is going to stay there. So let me see if I can get rid of that. Okay. Back in business. All right, phylogenetics and conservation. So phylogenetics is the recovery of evolutionary histories, creating phylogenetic trees. Molecular phylogenetics uses genetic information, mostly, to recover evolutionary histories and create a topology, which is the branching patterns. And you also get branch lengths, which tells you how much divergence there's been. One suggestion to get phylogenetic diversity is to select representatives from certain lineages. So, for example, if you have a field with several asters, several orchids, and what's another species of plant? So asters, orchids, and let's say honeysuckles, that you don't grab 3 honeysuckles, you don't get 2. If you could, you don't get 3 asters or 3 orchids. What you do is you select an orchid, an aster, and a honeysuckle. And that way you have representatives from each lineage. I mean, that would be ideal if that ever happens in reality. I don't know how that could come about, but when it comes to preserving diversity, what's called cryopreservation. So that would be freezing things. If you have a limited budget, you could only grab, say, seeds of certain plants, right? Or get the gametes of certain organisms. And you have a limited budget, you would grab one from each lineage to preserve that phylogenetic diversity. It also allows you to identify species-rich lineages, and those you might prioritize because if you are species-rich, it suggests you have some sort of genetic mechanism that allows for rapid adaptation. And that might be good if you are living in times of rapid environmental change. Okay, DNR barcoding. This is another use of DNA. So use markers that identify individual species. For most animals, it's cytochrome oxidase 1, so CO1. Um, and what that means is that is a marker because it is conserved within a species, but variable among species. And what you can do is grab cytochrome oxidase 1, sequence it, and look up the sequence, and it'll tell you what species you have. For plants, it's a little bit different uh, because the way that plants evolve and the rate at which it evolves, those genes evolve, you often need to get 2 or 3 different genes to get good species information, because chloroplasts will actually move between species much more readily than mitochondria do. Why would you use this? Forensics. So here's a caiman in Belize. It's protected. And if you can imagine, if somebody grabbed this and made a boot or a wallet out of it, which is illegal, um, you'd be able to use DNA barcoding to identify what species of caiman produced that boot or that alligator. So wildlife biologists will use this. And I mentioned APHIS, APHIS, and they use these techniques to look at animals that may have been imported illegally, so they can identify what they are. They've also used DNA barcoding to identify food sources. So I've seen where it was actually made the front page of the New York Times. A high school teacher and his students went into some sushi bars. They grabbed some sushi and, excuse me, and then they identified it down to species, and it wasn't the real high-end fish that they were marketing. It was actually some cheap fish, but they were charging the price. I also talked to a gentleman that sampled meat, and they were selling this ground meat as beef in several countries, and it ends up being horse meat. And what's interesting about it is some countries thought that was a better deal than getting beef. DNA barcoding is used in ecology to identify hard to identify species. So my lab is using it to identify insects. And I've seen DNA barcoding used in another forensic sense would be when airplanes collide with birds, that can use barcoding to identify the type of bird that collided with the plane. Okay, so DNA barcoding is targeted, in which case you use a primer for a group of species, and you get one species back. So it only amplifies a single species. There's something called eDNA, and you can use next generation or DNA barcoding or metabarcoding to identify suites of species. But eDNA, just sort of a strict definition, is when you look at DNA that's shed into the environment. And I've seen papers where they use eDNA. So you take a sample of water, you spin it down, and then you use sort of these fuzzy primers or imperfect primers to identify what species are there. And so you can say these mammals are found in these bays and not these bays. You can monitor biodiversity in and on soil. So soil and vertebrates. I've also seen um, read a paper where they looked at insect DNA, and what they did is they put traps out to collect insects, and then they just sampled the ground. So an insect dies, it goes on the ground, and as its cells break down, the DNA kind of spreads all over. It ends up that if you just sample the soil surface, you can actually sample what insects are in the area. So you don't even need to go out and collect insects. You just grab a soil sample, and it gives you a good index of what's out there. The other cool thing I've seen about eDNA, and we have a Wilkes student working on this. So she looks for snow leopard poop and then grabs the DNA and then can identify species. And what that allows you, identifies individuals. And what that allows you to do is to estimate the population size and also the relatedness between the individuals that you sample. So next generation sampling more broadly is when you use these imperfect primers to amplify DNA several species at once. So barcoding, you get a single species amplified. Even if you have contaminants, let's say you have 3 insects in your sample and you grind them up unknowingly, usually after so many cycles in your thermocycler, only one of those is going to win and be detectable. Next generation sampling, what you do is you use these primers that are not so good. And so everybody is amplified at low levels. And what you do is that you use those samples to figure out all the things that are there. So you can actually get a whole mix of sequences, which is really cool. Use that with highly diverse species samples. So a microbiologist that was at Wilkes was using it to look at the diversity of soil bacteria. And I've also seen where next generation sequencing is used to analyze diet. And what they did was you actually put a band around the bird's neck, loose enough so they can breathe, but tight enough so they can't swallow food. And these are chicks. And then what you do is you go in with a pair of forceps, you pull out the bolus, which is a little ball of food. It'll be filled with insects. And then you can identify what insects are important for that species. So you know how best to conserve that species. And that appears to be the last slide. So we are done. And that's it for conservation biology. It's a great semester, a little bit hectic. And if you have any questions, please feel free to email me.